Good morning, everyone. I guess it's mid-morning now. Sorry. So this summer, I had the opportunity to work under Nicole Lewandowski and Kristen Summer, looking at carbon cycling and seeing how calcium carbonate from coccolithophores might be affected by increases in anthropogenic CO2, meaning carbon dioxide that has been derived from humans. So first, I want to start us off with a atmospheric carbon dioxide timeline. We're going to start in the pre-industrial era before we start burning fossil fuels. At this time, we have a 280 parts per million atmosphere, meaning that for every million particles that we have, 280 of them are carbon dioxide. So we're going to fast forward to our present day, where we are now at a 400 parts per million atmosphere. This is only 250 years later, and this is higher than it has been in the past 800,000 years, for some perspective. We're going to fast forward even more to end of century projections, where we have a 900 parts per million atmosphere. These are future projections if we don't get our act together and we don't put in sanctions for carbon dioxide. At this time, if I make it, as my uncle did, I will be 104 years old, and over my lifetime, I will have witnessed a 140% increase in atmospheric CO2. This is larger than the entire increase from the pre-industrial to present day, which was only a 47% increase. So as we can see, there are going to be drastic changes to our environment. And the really scary part is that these don't reflect actual emissions by land, as we can see that a third of these go into the ocean, as the ocean is a carbon sink, where we know that it disrupts processes, natural carbon cycling processes. And we can see this change, we can see this process called ocean acidification, as carbon dioxide comes in from the atmosphere and is dissolved in the surface ocean. And we know that this process, ocean acidification, affects organisms that naturally cycle carbon throughout the ocean from the surface to the deep sea. And a very important organism that we're going to be talking about is one that I studied, coccolithophores. So coccolithophores, they are a calcifying phytoplankton, which is a microscopic photosynthetic organism. And you might not be familiar with the name, because it is very long. However, I want you to think back to your Geology 101 course, okay? You remember that calcareous limestone? That's what these guys primarily make up. And I want you to think about the White Cliffs of Dover, that massive formation. That's primarily coccolithophores. And so we can see with these guys the potential they have to sequester carbon from the atmosphere into the sediment, and why they're so important in this carbon pump. And so that's what I've been looking at this summer, this carbon pump. So we have, as I mentioned before, carbon coming in from the atmosphere, dissolving into that surface ocean, being used by all kinds of phytoplankton organisms for photosynthesis and for making shells in calcium carbonate organisms. And we have that moving to the ocean interior, and that is the biological carbon pump. And so what we initially thought at the beginning of this research is that when we increased anthropogenic CO2, we would see an increase in ocean acidification, that process that hinders coccolithophores and calcifying animals' ability to make shells. So we thought that over these simulations, we would see a decrease in overall carbon export into the deep sea through calcium carbonate, which we're going to refer to as particulate inorganic carbon. All right. And so here's our research question again. We looked at how the exchange of calcium carbonate might differ in an acidified ocean. And so in order to do that with data that might not be available to us, like pre-industrial era or in the future, we used what's called the community Earth system model, the community Earth system model, which is an Earth system model housed here at NCAR, not here, but there. Anyway, yeah, so in order to, so let me back up a little bit. So the community Earth system model, what it does is it's several models that'll run, they can run by themselves, to show different processes. Like ocean, we're looking at the biological carbon pump. Different processes happen on land, different processes in the atmosphere, land ice, sea ice. However, they can be coupled together through a few lines of code. This coupler is more than a few. But anyway, so we're putting these processes together to see what happens when we look at the Earth as a whole system. So what we're going to focus on in this study, specifically to see the effect of a certain amount of carbon in the atmosphere, is we are going to couple we're going to use our ocean and sea ice together. And then we're going to have these forcings of 280, 400, and 900 parts per million. Pretty much we're just setting the atmosphere at this level, and we're not letting it change. And so further to see our coccolithophores and how they're doing, what we're going to do is look at the different phytoplankton in the model. However, this is, excuse me, let me back up. Previously, in other CSM models, in the CSM model, there were only 3 groups of phytoplankton, calcifiers being only a small fraction of the small phytoplankton. This is not advantageous to the carbon pump because they are so important in moving material from the surface to the deep sea. So we use this really awesome new model developed by Kristen Krumhart, one of my mentors. And we saw coccolithophores now being their own explicit group, which means instead of being a small phytoplankton and only seeing small percentages change, we're able to see how an overlying carbon or differing carbon in the atmosphere might affect this group. Okay, sure. So this is now some output from the model. As we can see in the high latitudes, this is where we have high flux of calcium carbonate. This is where there are a lot of coccolithophores. And it matches up with satellite data. However, in order to get accuracy for what I'm doing and for what we're doing, to see at depth what changes, how the export changes, we are, so this is a plot showing, or an animation showing, that we made showing the change in depth with calcium carbonate. As you can see, the numbers moving up top are the meters as we go down. And we can see the blooms in the spot that we saw in the previous slide. So we've hit the maximum. We can see topography showing up at the mid-Atlantic ridges and then steep ocean. So in order to verify the model at depth, as we want to do, we use sediment trap data from the Bermuda Atlantic time. So this is, and we use these sediment traps, which pretty much just collect carbon particles. We use particulate organic carbon due to the unavailability of particulate inorganic carbon. However, we are able to convert this to particulate inorganic carbon through what's called the rain ratio. And we validated this with the model with 2 different rain ratios, a very low estimate of calcium carbonate through the Sarmiento et al. 2002 study, and then another one, the Cove 2002 study. And we can see the Sarmiento et al., because it is a more general Atlantic study, we see a very low end, and Cove, because it is at the bat site, you see a very or a higher end. And what I want to point out is that the model and the real life measurements are on the same order of magnitude, so it works to a good extent. And so we're going to continue on to actual results. So this is really interesting. We here see, I want to show you from the pre-industrial in magenta to the present day in dark blue, we see an increase in calcium carbonate flux. And what I want to tell you guys today is that is because coccolithophores are actually naturally limited in carbon that they can take up for photosynthesis. So when you have more carbon in the ocean, what happens is, sorry, something weird. So what happens is they're able to increase their biomass, which turns into more calcium carbonate export. And so we can see from the pre-industrial to present day, a very, not a very large, but a slight increase in calcium carbonate being produced. And then what we see from 400 parts per million to 900 parts per million is a rapid decrease, or a very large decrease, in this amount of calcium carbonate produced. And at this time, we believe that they are not able, coccolithophores are not able to calcify because of ocean acidification. And we can see this in another plot. So these are the regions where we see calcium carbonate increasing. So we have some spots where we have increased calcium carbonate flux, like off of Japan and China, up in the North Atlantic, the Southern Ocean, and off of Argentina. However, I want to show you that the net is a decrease in calcium carbonate, as we have shown in the integrated figure before. And so, as we've learned from this study, from the pre-industrial to end of century, what will happen is we have an increasing biomass due to less carbon limitation. So they're able to get more carbon, they're able to photosynthesize better. However, they are less able to calcify. So we're going to have less calcified cells, which will in turn create less calcium carbonate export. And so to return to our question, yes, we do have an effect from ocean acidification. However, very interestingly, we will also see an increase we also have seen an increase. This has also been validated by studies that show an increase in coccolithophores over the past, I believe it is from 1990 until now. And then we have a migration of coccolithophores towards the poles. However, it is predicted that in future studies we will see this effect of ocean acidification. And so this is our revised hypothesis. So as we have just stated, an increase because biomass in calcium carbonate export, then a decrease. And so what I would like to leave us with is that increasing carbon in the atmosphere will lead to our carbon sink of the ocean becoming less effective because it's not able to take in as much carbon. However, this is the 900 parts per million is a very scary ratio, or a scary concentration. However, if we are able to put in government sanctions, we can possibly get this down to the 600 parts per million scenario. And that's a lot better than 900. So I also want to say that this is our generation, and this is our time to fix this problem. Because if we won't, no one will. So I need to leave you guys with that, to think about carbon in your everyday life. All right, and many thanks to everyone here at UNAVCO. You guys have made this an amazing summer and made me think about things I never ever would have thought of before. Pride of thinking light, right? And I want to thank my mentors, Nikki Lewandowski, Kristen Krumhart, and Derek Weller, my communications mentor, for their help in guiding me through MATLAB and biogeochemistry, as it was a struggle. And everyone here, you all have been so advantageous to my learning environment and just personal growth and development. My cubicle buddy, Holly, and all of the Yusuf and your Launchpad and RESESS interns. Thank you guys so much. And I will now take questions.
Excellent. Thanks, Eddie. First off, great talk. Really interested to see what you're doing here. So my question relates to the setup of your model that you briefly talked about back at the beginning. With the CSM model, you chose to incorporate the ocean with the sea ice. Could you elaborate why you chose to include the sea ice component?
Unfortunately, I cannot with any accuracy. That is not necessarily something I looked into too much. I was more focused on just I focused more on kind of the ocean component.
Mm-hmm.
Sorry about that.
Well, could you speculate on perhaps what decreased sea ice in the future would mean for the exchange of atmospheric and oceanic CO2?
Huh. Okay. Well, as someone who's only been studying this for 11 weeks and has no background in oceanography, I will give it my best shot. However, we've got an input of fresh water into the ocean, and that is going to change stratification. So what we're going to have happen is more stratified layers and an inability for coccolithophores to do better. That's my general understanding. Yeah. So that's my understanding so far. Yeah. Thank you.
Oh, it was Glenn first?
Oh.
I'm already down here. Yeah, I skipped Glenn.
Yeah, my question, obviously at some point, sometime after 400 parts per million, but before 900 parts per million, you reach a peak sequestration, and at which point you start to decline, perhaps rapidly. I know it's hard to draw a curve between 3 data points, but from other information you've been working with, do you have any sort of indication about where that peak might be? Is it 600 or 800 parts per million, or?
I do believe it is at 600.
My question, I guess, is sort of related to the previous one, related to the freshwater input.
Yeah.
I don't remember everything that I learned in chemistry, but
Of course.
if you're going to make calcium carbonate, you need calcium as well as CO2. So how did you deal with calcium in the model?
So in oceanography, when we're looking at concentrations of calcium and salts in the ocean for calcification, we assume that there is an abundance of calcium. As we have seen that the limiting factor is the carbon in several studies. So we do see this limiting carbonate in previous studies and laboratory studies. So we assume that calcium is abundant all over. Yeah. Does that answer your question? Okay.
Okay, I was going to say, sure, for a quick one. There was a hesitation. Well, maybe you can touch base with Addie at lunch. Okay. So thank you, Addie.